remains were discovered in a forest in Alam earlier this month. The victim is believed to have been dead for several months. However, investigations are facing one major problem. They cannot accurately determine the time since death.
When the body no longer provides answer, one team steps in.
By studying the type of insects present and their stage of development, forensic entomologists can provide critical information, such as time of death, whether a body was moved, possible cause of death,
no rigor mortis, and no liver moitis, and even the body temperature is no longer useful.
And insect activity is limited. So how do we estimate the postmortem interval now?
Okay, I have the answer. A recent forensic study may have the answer.
What did they find?
The DNA discovered that bacteria and fungi continue changing throughout decomposition, and these microbial changes can help estimate time since death. This study investigated whether microbial communities could be used to estimate the postmortem interval. In simple terms, can microbes act as a biological clock after the death? The researcher focused on two major microbial groups, bacteria and also fungi. They wanted to determine whether this microorganism remained useful even during skeletonization.
The researchers used 6 pig carcasses placed in a forest environment in Switzerland. The decomposition process was monitored for 149 days. Oral swab sample was collected, grass soil sample was collected. Samples were taken throughout different stages of decomposition.
The researchers then used DNA sequencing techniques to identify bacterial and fungal communities. Microbial DNA successfully extracted. Sequencing analysis initiated.
The microbial data was then analyzed using machine learning models, and this allowed the researcher to predict the postmortem interval based on the microbial succession.
The study found that microbial communities change in predictable patterns over time. During decomposition, bacterial communities change rapidly. This made bacteria especially useful during the early stage after death. During the decomposition stage, fungal communities become more dominant. Fungi remain useful even during skeletonization.
When bacterial and fungal data were combined together, the prediction accuracy improved significantly. The microbiome model estimated the postmortem interval with a very low prediction error.
So, why is this research important in forensic science? In highly decomposed bodies, traditional forensic methods often become unreliable. Fingerprints may disappear. Insect evidence may no longer be present.
And body temperature can no longer estimate time since death.
However, microbial communities continues changing for weeks and even months after death. This allows forensic scientists to estimate postmortem intervals more accurately.
Determining the time since death is extremely important. It helps investigators verify alibis, narrow down the suspects, and reconstruct criminal investigations. These studies demonstrate that the microbes may become an important form of forensic evidence in the future.
Although the body decomposes, the microbial communities continue changing over time. This research demonstrates how forensic microbiology may improve postmortem interval estimation during advanced decomposition. In forensic science, even microbes can help reveal the truth. Every tension tells a story. Sometimes the smallest evidence speaks the loudest.
This paper, titled Dual Kingdom Necrobiome Successions, Extend Postmortem Interval Estimation into Skeletonization, was published in 2026 in Forensic Science International Genetics. The research was conducted by Elie Pascolo-Thiesche and his team from the University of Bern, Switzerland. The main concept of this study is that bacteria and fungi change in unpredictable patterns during death, allowing scientists to estimate the postmortem interval even during advanced decompositions and skeletonization when traditional forensic methods are no longer effective.